Hi, so let's discuss the different culture medias that we use in microbiology lab. So the very first being the conventional culture medias. So here we have the basal and the simple media. This is the first type, basal or simple media, in which we have the peptone water, nutrient broth, nutrient agar, and we have the semi-solid media. Now, in peptone water, we have peptone 1%, water, and NaCl 0.5%. Then in nutrient broth, we have peptone water plus we have the meat extract, which is 1%. Then in case of nutrient agar, we have the nutrient broth plus 2% of the agar. Then in semi-solid media, the agar percentage is reduced to from 0.2 to 0.5%. Now, this basal simple media is used for the growth of the non-fastidious bacteria. The nutrient broth is used to study the bacterial growth curve. The nutrient agar is used for the various biochemical tests like catalase, oxidase, and so tests. Then this semi-solid media is for checking or studying the motility of the different bacteria. Now, on number two, we have the enriched media. Now, enriched media will have something special. Like the culture will be enriched with something like it can be blood, it can be serum. Okay, so it will be enriched with something. So in this, we have the blood agar, the chocolate agar, Loeffler's serum slope. Now, see, in blood agar, what do we have is we'll have the simple basal constituents will be there. The media simple one will be there, but in that, we'll be having 5 to 10% of the sheep blood. Now this is heated to 45 degree Celsius approx. Okay, so in this we basically check the hemolytic property of the bacteria, like whether it is completely or partially hemolyzing the like the blood cells, right? So it is basically alpha or beta. Alpha is for the partial hemolysis and beta is for the complete hemolysis. So hemolytic property of bacteria is checked from this blood agar. Now, in case of chocolate agar, it's again 5 to 10% sheep blood only, but here the blood is heated to 70 degrees Celsius. So, what happens is that the RBCs are lysed and the content inside the RBCs are released, because of which it changes the color from bright red to dark red or brownish type of color. Okay, so in this, we basically grow a very fastidious bacteria which is Haemophilus influenzae, right? Then comes after blood agar, chocolate agar, we have the Loeffler serum slope. In Loeffler serum slope, we basically use the serum. So here we grow Corynebacterium diphtheri, which is also a fastidious bacteria. Now, the third type is the enrichment broth. Now, you need to make it very much clear that it is a liquid media. Broth is liquid, right? So, enrichment broth will be a liquid media that basically it selectively allows the growth of few microorganisms, okay, like few bacteria, and it will not allow the rest growth of the rest of the bacteria. So basically, when you have stool samples, you have sputum, so you have certain normal flora also. So it will not allow the growth of the normal flora, and it will only allow the growth of the pathogenic bacteria. Okay, so selectively allowing the growth of some bacteria. So here in enrichment broth, we have selenite F broth, we have gram negative broth, we have alkaline peptone water, we have tetrathionate broth. Now tetrathionate broth is for Salmonella typhi. The selenite F broth and gram negative broth are for Shigella, and this alkaline peptone water is for Vibrio cholerae. Now, next we have the selective media. The selective culture media is a solid culture media in which selectively allows the growth of some bacteria and while not allowing the growth of the others. So it's a similar to enrichment broth, but it is a solid culture media. So in this we have the LJ media or the Lowenstein-Jensen media for Mycobacterium tuberculosis. We have the TCBS, which is thiosulfate citrate bile salt sucrose agar for Vibrio cholerae. We have the DCA and XLD, which is DCA stands for deoxycholate citrate agar and XLD is xylose lysine deoxycholate agar. This is for Salmonella and Shigella, both of them. Then we have the PTA, which is the potassium tellurite agar, which is for Corynebacterium diphtheri. Then next we have the transport media. So transport media is used, suppose there's a delay in the between the sample collection and the examination. So if there is a delay, or if we suspect that the sample may contain some delicate microorganisms, so we use the transport media for that. In transport media, the bacteria are not growing, they're simply viable. They remain in the viable state, but they are not growing. So in this, for Vibrio cholerae, we have the VR media, which is the Venkatraman Ramakrishnan media. Then we have the Carrie Blair media. Okay, then for Salmonella and Shigella also we have Carrie Blair. ठीक है, media. And for Neisseria, we have the Stewart's media. Then comes the differential media. So differential media may we basically, as the name suggests, differential. That is to differentiate between the two groups of bacteria. So we have the McConkey agar and we have the Cled agar. So McConkey agar basically differentiate between lactose fermenters and non-lactose fermenters. The ones those who are lactose fermenters will be producing pink color colonies, and the non-lactose fermenters will remain colorless. So basically, here in this McConkey agar, the lactose fermenters, like suppose example is Escherichia coli, non-lactose fermenters, suppose example Shigella. Okay, now what is the composition? That is very important for McConkey agar. You can remember it with a mnemonic plant, that is peptone. Then L stands for lactose, A stands for agar, N stands for neutral red, which is indicator, and T stands for taurocolate. Then the second type is the Cled agar, which is cysteine, lactose, electrolyte deficient agar. Now, in this also you differentiate between lactose and non-lactose fermenters, groups of bacteria, but here it is used for the urine sample. Then at last we have the anaerobic culture media. The anaerobic culture media के अंदर we have the example that is Robertson cooked meat broth, which basically contains beef meat particles, meat extract, okay, beef heart, and it is basically it has glutathione and unsaturated fatty acids. Okay, so basically it is for the growth of the bacteria, which basically requires anaerobic conditions. Thank you.